to use chemical synthesis of making DNA, and each individual step of this process is around 99% efficient. Now, when you guys were in organic chemistry, you would have loved it if you had a process that was 99% efficient, like this process is. So, extremely efficient and good for making long stretches. Now, I'm just showing you a short stretch here, just because you'll get the point once we do a couple. But there's a specific process. Okay, so it starts with a controlled pore glass bead, CPG bead, and chemically, when you're making DNA, it's made in a 3' to 5' direction. Now, if you're making DNA enzymatically, when DNA is made by an enzyme, the enzyme makes it in a 5' to 3' direction. But the chemistry just worked out better to make it in a 3' to 5' direction, the opposite direction. And it's almost indistinguishable, except for there will be a phosphate missing on the 5' end. So it's almost indistinguishable from enzymatically made DNA. It's except for that missing 5' phosphate on the very end, it would be completely indistinguishable. So this is in your lab manual. What I'm going to show you is on page 96, 97, 98, etc. of your lab manual. So there's a couple things which you're going to use when making oligonucleotides. And one of the things, as I've said, is a little controlled pore glass bead, CPG bead. And attached to that CPG bead is a linker, just to give some space between the base that you want to, the first base at the 3' end of the sequence that you want to make. So you have a CPG bead, and attached to that CPG bead is, in this case, because our sequence ended with a T, so attached to the CPG bead is a linker, and then there's a T on it in the place of the B, which just stands for base. And there is a protecting group on here, a dimethoxy triphenyl group that is protecting that very reactive oxygen on that 5' end after this linker. And we're going to be adding phosphoramidites, one after the other, each phosphoramidite having the next base in the sequence that we want to add. And the phosphoramidite itself has a bunch of protecting groups on it. It has groups protecting the phosphate, and it also has, each individual base has the next dimethoxy triphenyl protecting group, again protecting that very reactive oxygen. So what's going to happen here, and let me show you a bigger picture of the DMT. So this is just a blow up basically of the DMT group, so showing you how there's steric hindrance around because of this DMT group, protecting this oxygen right there, and until the DMT group is removed, nothing's going to react with that oxygen. And again, this is all in your lab manual. So, the process for making oligonucleotides. You start off with your first base, so the CPG, the linker, the first base added to it. And you need to start reacting this oxygen, so you need to, as your first step, get rid of that DMT group. So trichloroacetic acid is added, which is a process called detrotylation, and that removes that DMT protecting group, so now this oxygen is ready to react. And then you're going to add a phosphoramidite, which contains the next base in a row. So in this case, the next sequence that we wanted next to the T is a G. So we wanted to add a G next to the T in our sequence. So this would be a G right here, and this incoming phosphoramidite would react with tetrazole in what's called a coupling reaction. And so this is your T right here, your 3' base, and this would be your G right there, more 5'. And now you can see it's starting to look a little bit like DNA would look, except that you have this trivalent phosphate, which does not quite look like DNA yet. So to convert that trivalent phosphate into a pentavalent phosphate, you're going to add iodine, which makes it undergo an oxidation reaction, and now you're starting to get a pentavalent phosphate, again, still with this protecting group to keep it from reacting with anything else. And so now, on this DMT right here, that's protecting it, and again, it has to be removed with TCA so that you can get the next base in a row reacting. So, in this scenario, you are assuming absolutely perfect addition of bases, but remember it's only 99% efficient. So, what happens if you have something that's only 99% efficient? Well, there is a strategy for dealing with that. So, let's say the next base you want is a C. And so let's say the C adds here, and the C adds here, but the C failed to add right there. If you don't have a way of coping and stopping the addition from anything else, you're going to, at this point, start getting incorrect bases added. And again, I've only drawn in, like 3 T's right there, but each little bead is coated with hundreds of little T's. So when I say a 99% reaction, you don't only have 3 DNA strands being made, you have thousands and thousands of DNA strands that are actually being made, and potentially some of them could be incorrect if you don't have a way of dealing with that. So let's say that C temporarily does not get added, but let's say the next T in a row does. And then there's a C after that gets added. And now let's say, we want to add a G, but let's say the G gets added here, and the G gets added here, but the G does not get added to this one. So now we're missing a C in this one, but we're missing a G in this one. And let's say the rest of them get added just fine. So G-A-C-T-A, G-A-C-T-A. So if you wanted to use these oligos to bind to DNA, if you had an incorrect sequence here, they're potentially going to bind to an incorrect place on the DNA. So how are we going to handle that problem? Well, there is a way. And that way is after the phosphoramidites and the tetrazole are added, acetic anhydride is put through. And if the phosphoramidite did not properly add in its proper place, the oxygen is still reactive. And if you put acetic anhydride through, then that oxygen is capped and no longer reactive. But if it got a phosphoramidite properly added on there, then that oxygen has reacted with this phosphoramidite, and this oxygen is protected by DMT, so putting that acetic anhydride through has no effect if everything has been properly added. So what you get is you get detritylation with TCA to remove the DMT, you get phosphoramidite in the presence of tetrazole in a coupling reaction, then you get acetic anhydride put through in a capping reaction, just in case it stops the growth of any of those incorrect strands. Then you get oxidation with iodine, and you do this cycle again and again and again until you have something that is the correct length. And then you go to deprotection. So in your lab manual, you have this picture in your lab manual right there. This is the important thing to know for the exam right here. So there are 5 steps in the procedure. As I've said, deturylation, addition, capping, oxidation, and the 5th step is deprotection. This is in your lab manual. So for the exam, you should know the names of the steps, deturylation, addition, capping. You should know the chemical that's used to carry out those steps, and you should know the purpose of that chemical. So what is the purpose of iodine, what is the purpose of acetic anhydride, etc. And in the 5th step, the deprotection step, you're adding ammonium hydroxide to it, and that ammonium hydroxide is going to remove the protecting groups from the base. It's going to remove your strand of DNA from your CPG bead and the linker, and it's going to remove the DMT from that final group. So what you're going to end up with at the end of it you're going to end up with basically the correct sequence, but remember, enzymatically it goes this way, chemically it's going that way, and so the only difference is it's going to be missing a 5' phosphate because of the way the chemicals added on in terms of the phosphoramidites. So, but that missing 5' phosphate in itself can be helpful. If you need to radioactively label something, you can just use a phosphatase, an enzyme which is going to add a phosphate onto it. You can put some radioactive phosphate onto it, and then it's going to be radioactively labeled very, very easily. Okay, so how are we going to deal with this incorrect sequence? Well, remember that capping step, if it's capped, it's no longer adding onto anything which is capped. And so the only thing which will be the correct size will be the one with the correct sequence. And if you separate it on an HPLC column, like a C18 HPLC column, it is very easy to separate a full-length DNA fragment from much shorter fragments. So after they make the oligonucleotide, they purify it and would send you basically just the full-length fragments. Now, the company, the #1 company in the United States that makes oligonucleotides is a company called IDT. And I will admit, I'm a fangirl for IDT. I did a company visit there. They're in Coralville, Iowa. They're also up in Carlsbad. They hire people up in Carlsbad to make oligonucleotides because this is a big biotech area. And so when I visited the company headquarters in Coralville, Iowa, I was amazed at the streamlined ability of the company. So when you get on the computer to the IDT website and you order your sequence, you put in what sequence you want for your oligonucleotide, and they're a lot less expensive because they've automated almost the entire process, so very few people are actually needed in this process. So you order a sequence, and it automatically generates a barcode that somebody then just sticks onto and puts it into a little rack. And these are like 96, I don't know whether you guys have ever seen 96 well plates, they're about this big. So imagine 96 little holes in there, each one having a tiny little tube about this high. And inside each of those little tiny tubes are a bunch of these CPG beads with the first base on there. So a computer basically is telling a minimum wage worker, here's a barcode, you know, grab something starting with a T, slap this barcode on it, put it in a rack, put it in the machine. And so that you just need a person to sit there and do that all day. So very little education in terms of that. Most of their money goes into research and special labeling of stuff and things like that. So basically, all of their machines operate on that barcode. That barcode is scanned by the machine, so each machine is doing 96 different oligos all at one time. So 96 different chemical reactions. This tube needs a C next, this tube needs a G next, this tube needs an A next, etc. And it's able to detect, it's measuring the inflow and it's measuring the outflow of all the nucleotides. So it can tell if there's a problem with the addition, If a significant amount of the oligo is not being made properly, so for example, let's say instead of 99% reacting, let's say only 5% react. Well, it's going to measure and say 95% of my phosphoramidites have come out the bottom and have not reacted. This says there's something wrong with this tube. It will automatically send a note to the computer to start that one again. So somebody else can stick, the next barcode on it and start it again. And the computer keeps track of where that new barcoded one is. So everything's computer controlled. Have you guys seen the Indiana Jones movie where he's got the Ark of the Covenant and he's sort of wheeling it into that big warehouse at the end, and you just see boxes and boxes and boxes? The warehouse at IDT is like that. It's a big warehouse, and you look across the floor, and there's row after row after row of machines, all making, each machine is making, so the machines are like maybe like this, like that. Each machine is making 96 oligonucleotides at a time. So they make tens of thousands, like 100,000 or so, different oligonucleotides every day and send them all over the world. So it's a huge business because a lot of people in their research are using oligonucleotides. Police departments use oligonucleotides, research facilities, companies, everyone's using oligos, and this is a very efficient process for how they get made. So the only thing that's not automated, and after the ammonium hydroxide is added automatically to each one, and then you have the output of each tube after it's soaked in the ammonium hydroxide for a certain period of time. The output automatically in the tube is dried down and sampled on mass spec to make sure it's the correct molecular weight. If it's not the correct molecular weight, it's automatically started again. Everything is automated in this company. It was beautiful. The only thing that's not automated is shipping. Because very often people will order, say, 20 different oligos at a time. Some get made on this machine, some get made on that machine, some get made over here. And so they needed a person to pull them all through, because otherwise they're shipping out 20 different envelopes and it was costing them money. And they said, it's worth it for us to pay, to have somebody look at it and make sure that we've pulled all 20 from all different machines and we can just set them out in like a single envelope. But all the time, people will order a whole bunch of oligos from this company that uses this particular process. So make sure that you understand how oligos are made if going for a job at IDT, or there's other companies in San Diego that will also make oligonucleotide. IDT just happens to be the largest worldwide, but there are other companies in San Diego that make oligos that hire people all the time, that hire people with bachelor's degrees, and in the interview you can impress them by understanding their business process, understanding how oligos are made during the interview. It will give you a leg up on people from UCSD who know chemistry, but not necessarily the ins and outs and the little details of things like this. Okay, so in this experiment, we are going to be doing PCR, and we're going to isolate DNA. So in this experiment, PCR of short tandem repeats, you're going to be isolating buccal DNA. Now, buccal DNA is your epithelial cells inside of your mouth. And so the overall procedure is there's going to be a protocol for how do I isolate DNA from our cells. And then you're going to be PCRing your DNA at 3 locations. And so there's a different protocol for how to do a PCR. You're going to have to put that together because they're 2 separate protocols. And then you're going to be running them, you're going to be splitting up your PCR on agarose and acrylamide gels, and you're going to be seeing how they look on both types of gels. They might, different types of gels will separate the DNAs a little bit differently, the size ranges. And then you're going to look at a gel with short tandem repeats on it. And you're going to, and I'll be explaining what a short tandem repeat is in just a moment, and you're going to be doing some statistics on that. So this is the overall process of the experiment that you're going to be doing, and you're going to have to put everything together. So I'm going to be talking about this sort of step by step. So the first thing that you're going to be doing is on page 127. So in your lab manual, page 127, isolation of DNA using the DNA IQ system. So if you turn to page 127 in your lab manual, you will see that you're going to start out by adding a lysis buffer to your buccal cells. So you're going to have a swab, and you're going to swab the inside of your mouth and collect a bunch of epithelial cells on that swab. And then you're going to put it in a test tube, and you're going to be adding lysis buffer. Now, you guys have already been exposed to lysis buffer. You've heard about, you've done a GST experiment where you use lysis buffer. You've heard about the plasmid experiment, so the components of lysis buffer. So basically, you are adding lysis buffer that's designed to break open the cells. And the only different thing is, in this case, it has DTT, diethiothriotol, which is going to help with breaking open the epithelial cells and sort of stabilizing the DNA a little bit. So you add DTT to the lysis buffer to cause the cells to break open and free the DNA. Now, in that lysis buffer, you have guanidine. And so this is a very important extra component of the lysis buffer. It contains guanidine. And after you have, and you could read the protocol on page 127, and it's just some very specific things, adding 250 microliters of the lysis buffer, transfer to a new tube, blah, blah, blah. You're going to be adding resin to your supernatant, your lysed open, freed genomic DNA. So you've lysed open the epithelial cells, and you have genomic DNA basically in the solution. And that solution, remember, also has guanidine in it. So you're going to be adding beads. So the resin that you're adding has an iron core in the bead. And that iron core, when put next to a magnet, will cause all of those beads to snap over to the side of the tube. And this is going to make it very, very easy to remove the supernatant and exchange buffers and do the wash and stuff like that. So rather than trying to pellet something down, rather than trying to pellet, a tiny little pellet down of these beads and trying to remove supernatant around it without disturbing the resin beads themselves, you have a magnet that slaps over to the side, and then you can just go all the way down to the bottom of the tube without worrying about disturbing any kind of a pellet. So you'll see it's a kind of a very different, kind of cool approach for how can we grab things on. So in the presence of guanidine, DNA will make a salt bridge with this little bead. And this little bead, it's a proprietary bead that they do not tell you what is on that bead that's going to make a salt bridge with the DNA. But they will say, in the presence of guanidine, DNA will make a salt bridge to the bead. And as long as you keep guanidine in the solution, your DNA will keep sticking to the bead. So you're going to add the resin in and move it over to the side and of course you're going to be adding more lysis buffer. You can read the protocol there. You're doing this a couple times, making sure you get all the proteins off of the DNA, stuff like that. And then you're going to be adding wash buffer, again, making sure you get all the proteins off of the DNA by washing. And every time you add a new buffer, you're going to put it next to a magnet. Those beads are going to slap over to the side so that you can very quickly exchange the buffer in the tube. So then eventually, after you've done all the washes, you want to be able to elute your DNA out of off of the beads. And so the catch here is now your elution buffer, if you put an elution buffer on there that does not contain guanidine, all of your DNA is going to come off of the beads, and then you can put those beads up against that magnet, and now you're removing your solution which contains DNA, but none of the beads, because the beads are all over to the side. So you're going to be reading the protocol as part of your putting everything together experience in the intro. So you're doing the buccal swab. You are doing the procedure to isolate DNA, and the oligos have already been made for you. The oligos are made outside, and they're included in the kit that you're going to be using. Okay, so this kit that you're going to be using relies on PCR. And just as a reminder, you should already know this from Chem 560, but you have a template strand, and you're going to have some DNA that's binding to the template strand. So you can see the black strand up there is your template strand. The blue strand would be your oligo that's binding to the template strand, the opposite sequence, and this should all be very familiar from 560. And in the presence of DNTPs and polymerase, the next nucleotide in a line comes in and is added by the polymerase, one at a time, moving down the DNA strand. So you all remember this from 560, right? Okay, just making sure we're all on the same page, that you've all had 560 and everybody remembers that basic idea before we get into the PCR. Okay, so PCR is polymerase chain reaction. And it's basically a way of amplifying short stretches of DNA. Very short stretches of DNA. And remember, you're starting with genomic DNA. So you're starting with millions of bases of DNA, but you're only going to be amplifying, 600 bases here, 400 bases here. Very, very short stretches of DNA. People will amplify maybe up to 1 to 2 kilobases of DNA, but you're not in any sense amplifying an entire genome worth of DNA. It's very, very short stretches of DNA. And so PCR, this method of amplification, depends on 3 steps. And so the first step is a denaturation step. And that's usually done at a high temperature. So it's denatured around 95 degrees so that any double-stranded DNA now becomes single-stranded. And after your double-stranded DNA has become single-stranded, you're going to have oligonucleotides that are designed to bind to very specific sequences on this DNA. So if this represents, say, an entire chromosome, maybe you have, one oligonucleotide which binds here, one oligonucleotide which binds here, and you want to be able to amplify this short stretch of DNA in between those 2 primers. So after you denature the DNA, you have an annealing step next. And that annealing step is where you're trying to get your oligos to bind to the DNA. Now, DNA has something called melting temperature, and if you are ordering an oligonucleotide from a company like IDT, they're going to calculate the melting temperature for you. And it's going to depend on what is the GC content, what is the length, and stuff like that. So there's a calculation involved in that, and they'll just tell you, they'll say, okay, the melting temperature of this short stretch of DNA that you've ordered is 50 degrees Celsius. And so if you want then to anneal to the target of DNA, you need to make sure your annealing temperature is a couple degrees, usually about 5 degrees below the melting temperature. So your first step, denaturation, is done at around 95 degrees. Your second step, Annealing is done about 5 degrees below the annealing temperature, and this is going to vary. This is going to vary on the GC content, and it's going to vary on the length of the primer that you are using. So an annealing temperature might be 45 degrees, an annealing temperature might be 75 degrees, because it can completely vary based on the parameters. Okay, so you give it some time for these oligos to anneal to the perfect sequence in the DNA, and then you add TAC DNA polymerase, Thermus aquaticus DNA polymerase, and this TAC DNA polymerase will then extend the DNA. So this one, it will just start extending the DNA. It doesn't know. It says, well, you ideally want to amplify between these two, but it doesn't know that. It'll just start extending and keep going, as long as time permits. The polymerase works generally at about 1,000 bases per minute. So if you have, say, 1 kb you're trying to amplify, you would allow the polymerization step to happen for around 1 minute for 1,000 bases. And if you let it go for 5 minutes, it's going to keep going for 5,000 bases. So generally you can control how much addition there is. But let's say both of these go completely to the end. So this one goes completely there, and that one goes completely to the end. And so that's your extension. So denaturation, annealing, and extension. So then you're going to do it again. You're going to do a second cycle, a second denaturation, and so now you have 4 strands of DNA. Now you have denatured all 4 of those strands of DNA, and you are again allowing oligonucleotides to bind to their respective positions in the DNA, and then the polymerase is going to become active again. Polymerase works well at about 72 degrees. So basically you're doing 95 degrees for denaturation, you're lowering it maybe 45 to an annealing step, and then up to, like, say, 72 degrees for the DNA polymerase to start working, and it does its job with, this one gets extended all the way, and this one gets extended right to here, because that was where that one started. So now you're starting to see right here and right here, you're starting to see your target size piece of DNA from this. You still have longer pieces in there though. So then you're doing this again. So you get denaturation and annealing and extension, and now you're starting to see more of your actual target sized fragments appear. And so what's happening is every time you do a round of replication, every time you have a, say, original piece of DNA, you can still get a oligo binding to that original piece of DNA and extending beyond, here, but that's only going to be multiplied arithmetically. So if you have 20 rounds of replication, you're going to have 20 times where this goes to the end. But these are your target sequence is being multiplied exponentially. So 1 to 2 to 4, to 8, etc. So by the end, you're going to have a lot more of your target sequence than you will of what you started with. So I'll show you that in just a second. I'm going to put these back here. So that looks kind of like this. So now you can see in only the 4th round of replication, and normally you're doing, say, 25 to 30 rounds of replication. Only the 4th round of replication, and now a significant number of the pieces of DNA are your target size piece of DNA. So if you run this on a gel, your predominant product is going to be your specific target sequence on the DNA. You might have some other longer pieces, but they're going to be far and away in the minority, and the majority of it will be your target sequence. And in only 25 to 30 rounds of replication, you can easily have 1 million. You can go from 1 copy to 1 million copies of your target-sized piece of DNA, assuming that you have enough nucleotides and your polymerase lasts and stuff like that. Because remember, polymerase is an enzyme, and in the protein purification lectures, you learned about how enzymes have a half-life, And so, you know, you have to have conditions, you know, long enough. So eventually your enzyme itself will die out sometime around 20 or 30, or if you add enough, 35 rounds of replication. So you're going to be doing PCR on your genomic DNA that you isolated. Now there are some conditions you need to think about in PCR. So in the PCR reaction, if you're putting a reaction together, you're going to have buffer in a PCR reaction because you need to maintain a certain pH. You need to have magnesium chloride in there for a couple reasons. One, you need to have magnesium in there to allow the polymerase to work. But the reason that magnesium chloride allows the polymerase to work is that divalent cation has, at least in the case of polymerase, that divalent cation has positive charges. And the DNTPs, nucleotide triphosphate, has negative charges. And so you have the DNTP, and you have negative charges, and you have the positive charges of the cation, and they form a particular shape, and that shape is what is recognized by the polymerase. So if you don't have any magnesium at all, you're not going to get any result from adding polymerase with no magnesium. Now, one of the things I had you pick up was a page on PCR. So telling about PCR, the reactions, some of the important components in the reactions. It's kind of an old article, so it talks about, when it talks about hot start, it's talking about it in terms of wax, which we're no longer using, and I'll distinguish that in just a moment. But in terms of the things that you're adding to a PCR reaction, it gives a good explanation of the things that you're adding. So you also need to have DNTPs, your 4 bases, because you can't add bases if you don't have them in the reaction. You also need to have primers, and your primers have to be specifically designed to bind to specific target sequences. And you all have template DNA. So not only do you have your oligonucleotide that's going to bind to template DNA, but you have your genomic DNA in there, which is providing the template for the oligonucleotides to bind to. You need to have some water in there just so everything's not too concentrated, and of course a Taq polymerase to carry out the reaction. Now, one of the big problems with doing PCR originally was mispriming. And mispriming is where you're setting up a reaction at room temperature, and you have your oligonucleotide, and you have your target sequence of DNA, but you're setting it up at room temperature. And so the binding is all about hydrogen bonding. So you can have your oligonucleotide binding to a perfect sequence, but you can also have your oligonucleotide binding to a less than perfect sequence, and it has maybe enough hydrogen bonding to sort of stick it together. So you have perfect matches and you have less than perfect matches. So how do you assure that your DNA is only, your oligonucleotide DNA is only going to bind to the area of perfect match? And though there are different procedures that people have used to do what's called a hot start. Now, the first thing they would do is they would try you doing DMSO. And dimethyl sulfoxide, I think. DMSO was okay. DMSO destabilized the bonding of all the hydrogen bonds, so that if you had a partial match, it was destabilized to the point it was not binding at all. But if you had a perfect match, it was slightly destabilized, but still there was enough hydrogen bonding to keep it together perfectly. But they came up with better solutions after that. So in the process of hot start, they said, okay, well, what we really need is we need all the components that are separated, so that until we reach a perfect annealing temperature, until we're over that perfect annealing temperature, we're not going to get any enzymatic activity. Because if you have an oligo binding to DNA and it's only a partial match, and you have the enzyme in that same tube with the DNTPs and everything else that's needed to make the reaction go, you can have the Taq polymerase starting to add DNA from the incorrect location, and then you're just going to get more and more and more of the incorrect sized base. So one of these solutions they started saying for this hot start procedure, they said, okay, we're gonna heat up our solution so that it's over our annealing temperature, so that anything that is less than a perfect match has no chance of being there, and only the perfect matches are going to survive when it's at its annealing temperature. And then we're going to manually add the polymerase. And if you are doing one tube, sure, that'll work fine if you only have a single tube to try to add a solution to. But a lot of times when people are doing PCRs now, they're doing 50 or 100 tubes, in an entire assembly. So trying to catch it all just at the point where it's heating up and trying to add the polymerase to all 100 tubes is kind of a royal pain in the butt. So that didn't last very long. So then they came up with this idea of, well, what we really need to do is separate out the components. We need to make sure that until it's over a certain temperature, all the components can't be in the same tube. So what they would do is they would add, say, half of the components together, and they would have a little wax melted on top of half of the components, and they would put the other half of the components on top of that little layer of wax. And then, when you heat it up to the first denaturation temperature of 95 degrees, that wax melts, and the first solution goes, the top solution goes plunking into the bottom solution, and then everything is mixed and ready to go. But here's the key: when it's doing that, it's now over the annealing temperature. It never gets below the annealing temperature. And if it never gets below the annealing temperature until the very end of the cycle, by which time all of your amplification is already done, if it never gets below the annealing temperature, then you're only going to, you don't have to worry about partial matches anymore, because the temperature is high enough to destabilize that hydrogen bonding, so it's not going to happen. But of course, adding that little bit of wax to all 50 or 100 tubes is still kind of a burden. So then, and this is the procedure that we're going to use, the company came up with this idea and said, well, the real problem is the polymerase. If you have the polymerase that cannot add nucleotides until it gets over the annealing temperature, then your problem is solved. You can have everything in the same tube, including the polymerase, and if you have the active site of the polymerase blocked with something, so in this case it's blocked with a little piece of a protein, so if you have your active site blocked with a piece of a protein so that your enzyme does not have activity until after it's over the annealing temperature, then this solves the problem. So we're going to be using polymerase that has a protein blocking the active site, and that protein will not dislodge from the active site until you have heated it to around 95 degrees, which is our denaturation temperature, for a couple minutes. And then that piece falls out, and then the polymerase is going to function normally, and then you're going to get that denaturation, annealing, and extension 3-step process, and you're going to get a proper addition of the oligonucleotides where they should be binding. So let's see, that's pretty much the same thing that I was going to tell you. Okay, so now you're going to then run it on a gel. So after you do the PCR, you're going to be running it on a gel. And there's different kinds of gels. So you might want to take a picture of this. If you want to take a picture of that, this would be a good time. So there's different kinds of gels. There is a polyacrylamide gel, and a polyacrylamide gel will separate things in the range of 10 to 800 nucleotides or base pairs. So things on the smaller side. Things which are in the size range are small RNAs, small DNA restriction fragments, sequencing products. On the other hand, you have agarose gels. And agarose is a somewhat looser matrix compared to the polyacrylamide gel. So it will separate larger fragments, but it works on basically the same principle, is that the DNA or the protein, in the case that you've already done with the earlier in the semester, the protein of the DNA has to get through this matrix solution, whether it's a very thin matrix in the case of polyacrylamide, very small holes, or in the case of agarose, just so many overlapping long strands that it sort of forms a matrix, but it's easier to get through, so it's going to separate larger sized pieces. So agarose gels separate between 300 and 50,000 nucleotides or base pairs. Things which fall in this category are the larger RNAs or the large DNA restriction fragments. And within polyacrylamide and agarose gels, there's two different types of gels. You can have a native gel. A native gel is where you want to keep the DNA or the RNA together. So you'd have double-stranded DNA, or you'd have RNA in its native configuration. And this is done with just a normal salt buffer, like a tris borate with some salt, or a tris acetate buffer, and everything is kept in its native conformation. On the other hand, you can run a denaturing gel. And a denaturing gel is when you want to have single-stranded DNA, or you want to have RNA that's separated on the basis of its size and not its native conformation. And so if you have a polyacrylamide gel and you want to run a denaturing polyacrylamide gel for DNA, you've already experienced denaturing polyacrylamide gels for proteins, but this is for DNA. So if you need a denaturing gel for DNA, you can add 7 to 8 molar urea, which is a chaotropic agent. And this chaotropic agent, the urea, competes for hydrogen bonding with the DNA and the RNA and causes them to become single-stranded and to lose their native conformation. Now, if you're running an agarose gel and you want to do a denaturing agarose gel, if you're doing DNA, you can add sodium hydroxide, basically putting DNA in alkaline conditions. When DNA is in alkaline conditions, it will denature. So you run an agarose gel that has a high pH, and your DNA will stay denatured. RNA is a little bit more difficult. RNA, you generally have to use something like methylmercury, which is extremely toxic and deadly even if you're not wearing the right type of gloves, or glyoxal, which can cause severe allergic reactions in people who are allergic to it, or formaldehyde, which is a potential carcinogen. So none of these choices are good. So if you have to run a denaturing RNA gel, you're probably using formaldehyde as the least bad choice there. But even that comes with lots of environmental health and safety regulations for working with that stuff. So you're running a gel. Get my next slide here. Where is it? Again, you have a picture of this in your lab manual. On page, I don't know, page. Page 94, 93, 94, of your lab manual. Okay. It's hard to sit and listen to somebody for a couple hours, isn't it?
No.
My butt gets sore too, in case you're wondering. Okay, so let's say you have some long pieces of DNA, 45,000 base pairs, 28,000 base pairs, you cut them with the restriction enzyme, so you have a bunch of different smaller sized fragments, and you're going to run them on an agarose gel. This is just a for example. So you run them on the agarose gel, and so remember you had two different sizes making a bunch of different sized pieces. And unlike what you did in the aldolase experiment when you ran the STS gel, in that one you calculated the RF because nothing ran before the dye front. When you are dealing with DNA gels, polyacrylamide or agarose gels with DNA, you cannot use an RF because it is possible for small fragments of DNA to run before the dye. It's not separating on the same basis anyway. That it doesn't have glycine in there, so that's not happening at all in the same way. It's not an STS denaturing gel at all. So please do not confuse the two. So let's say you added fragments from DNA piece A that was cut up and fragments from DNA piece B, and you ran them on the gel. And then you measure. You can say, okay, well, B started out here, and I can measure how far did this piece run, how far did this piece run, how far did that piece run, and I know what size they are. And you can make a graph out of that. Kind of looks like this. And again, this is in your lab manual. So there'll be a part where it's fairly linear if you're using sort of a medium percentage gel. And remember, one of the things you learned in your aldolase experiment, you can choose all different percentages of gels. And you choose different percentages of gels based on what size protein, in that case, you were needing to separate. And the same thing holds true for DNA that holds true for protein. If you needed to separate out, and you can see it's a little bit more difficult to separate out, say 220 from 350, they're at the smaller end of what can be done in an agarose gel. 350, 600, you're really not getting any linearity in here at all. If you needed to separate out efficiently a 350 from a 500, from a 600, etc., you might want to make sure it's more linear in this region. So you would choose a higher percentage gel, because a higher percentage gel is going to separate out the lower molecular weight pieces better. And it does that with higher percentage gel. Everything has a harder time getting through the gel, but the smaller the pieces are, the easier time they're going to have getting through the gel. So a higher percentage is going to allow you to separate out all those small fragments, where all the large fragments, 3,000 and up, they're all going to be just basically in a big lump at the top and not separated out well at all. On the other hand, say you needed to separate out efficiently 6,000 from 12,000 from 19,000. Well, in that case, you would use a lower percentage gel, because a lower percentage gel is going to allow you to separate out the higher molecular weight fragments better. Because when there's larger holes, everything goes through faster, and these smaller pieces are all going to go through poof all at one time, so they're not going to separate out very well, but you're going to get some separation in the larger sized fragments. So you can choose your percentage of gel based on what you need to be separating out, and it's very flexible. And this holds true for agarose, holds true for a basic acrylamide, holds true for STS page gel. It's the same rule for all of them. It operates exactly the same. Okay, so let's back a little meter. So in this experiment, you're going to be working with biological material, and I actually developed this is the experiment that I developed for the class, because I was very interested in forensic science. I don't know whether you guys know, but I teach forensic science here. And when I was getting into forensic science, and I'd taken a bunch of classes and stuff, I decided I needed more practical experience. And so to get practical experience, I took one summer off and I worked at a paternity testing lab. And I took another summer off and I did an internship with the San Diego Police Department, learning forensic biology from them. And so I put together the different techniques that they were using in the labs to make this experiment. So the paternity testing lab was doing the DNA IQ system that we're going to use with the magnetic bead technology, and the police department will PCR up short tandem repeats. So this all comes from biological evidence. And biological evidence can be blood that's left behind in a crime scene. It can be semen that's in a rape case. It can be saliva. In a lot of rape cases, the victim might have their breast licked or something like that. It can be urine. There was one case where this burglar, and you're going to break into somebody's house, you might be a little nervous. Well, this burglar was a nervous peer, and so he had a habit of peeing against the side of the house that he was going to break into. So eventually the police figured this out, and they were able to get his epithelial cells, because you shed epithelial cells everywhere. You shed them when you urinate, you shed them when you poop, you shed them in your saliva. So you are discarding, you shed them when you touch things. So they were able to get his epithelial cells and get a genotype basically for him. So there was another burglar who, as his calling card, left poop in somebody's living room. So it's kind of like a power play. So he'd be like, I was here, and he would take a dump in the middle of somebody's living room. But what he didn't realize is the police can carefully get DNA from poop. Now, keep in mind that your poop is what you've eaten. So you have DNA from your food in your poop. You have DNA from bacteria in your intestines in your poop. But on the outside of your poop, as it slides along your colon, you're picking up a lot of epithelial cells from your colon. So a very careful extraction of just the outside of the poop gives you an enriched layer of epithelial cells, and then they can get a human profile. You're amplifying bacteria and food, but remember, they're designing this PCR oligonucleotide, so they're only going to bind to human DNA. So it doesn't matter if you get other kinds of DNA in there, you're amplifying only the human DNA. So you're going to have a source of biological evidence. In your case, you're going to be taking epithelial cells from your cheek to be able to amplify those. So initially, there were 13 locations on the DNA that the police would amplify. And these locations on the DNA had quite a bit of research that went into them. You have in your DNA coding regions that code for a gene, and you have non-coding regions. And the non-coding regions of your DNA are free to mutate without killing people. And so over many years, your non-coding regions of your DNA has mutated, and repeating segments of DNA have come to be through, it's an enzymatic process called stutter. And I'm not gonna go into the whys and wherefores because this is not a forensic science class, but you will get repeating sections of the DNA. So one of the handouts I gave you shows a little sequence of DNA, and it shows you AATG, 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 AATG repeated a number of times. So that is a short tandem repeat. Short because they're usually 4 to 5 bases long. Tandem because they're right next to each other, repeated over and over and over again. So what they have done, scientists since the 1980s, scientists have been developing this methodology since the 1980s. And what they've done is they've found places on the DNA which have a great deal of genetic variation from person to person. So, for example, if you are looking at blood types, the vast majority of people are type O positive. And a heck of a lot of people are type A positive. So if you find very few people are type B or type AB. So if you find type O positive blood at a crime scene, that's not very discriminatory in terms of being able to rule out who committed the crime. So there was a need in police work to be able to get away from just simply typing the DNA, typing the blood, to how can we use people's DNA to get a genetic profile. So the research that went into this found places in the DNA that had variation among people, because just like blood types, if almost everybody is type O or type A, and you find type O, it's pretty useless. But if you are looking at a place in the DNA, and you can say, okay, well, a lot of people have 8 repeats, but some people have 7, some people have 6, some people have 9, some people have 10. And maybe 10% of the people have 6, and 10%, and 20% have 7, and 30% have 8 repeats, one after the other. But then, some people have, percentage have 9, and percentage have 10, etc. So now you're saying, okay, if we find 6 repeats at this particular location on the DNA, we can use this to rule out anybody that has 7 or 8 or 9, etc. So you can use statistics to do this, to say how likely is it that you would have 6 repeats on this piece of DNA that we are looking at. So initially, the police identified 13 different locations on the DNA, and they were able to get, using statistics, they were able to get and say, well, the chances of having say TPOX, TPOX, or the D5S518, there's maybe only a 10% chance that anybody would match this pattern. Or at D5, maybe there's a 10% chance that anybody would match that pattern. And so when you start applying statistics, if you've checked 13 different locations on the DNA, it's the probability of this times the probability of this times the probability of that, and that gives you a much lower probability. So when they started looking at these original 13 locations, they were able to fairly easily get the probability of anybody having the same pattern of DNA is 1 in 10 billion, which was great initially. But now, as the population of the Earth is over 7 billion and climbing, eventually you're going to have some defense attorney that tells the jury the population is, the probability of someone else having the same exact pattern is 1 in 10 billion. And the population of the Earth is almost 1 in 10 billion. Therefore, there is another person on Earth that has the exact same genetic pattern as my client, and that, ladies and gentlemen of the jury, is the person that committed this crime. And for the jury, it doesn't matter how likely it is that person happens to be in San Diego at this exact time, because juries don't understand science. And if you have a good lawyer who can twist the science a little bit, you can get juries to believe a lot of things. So the police department eventually said, crap, 13 is not enough, we're going to go to 24 locations of the DNA. So now they have 24 different locations that they test on the DNA, all of them doing PCR. So they are PCRing up every single one of these locations, and all in the same time, and all in a single tube. So they have advanced the process so that they can do PCR on 24 different locations in the crime lab. They will isolate a sample of DNA from a crime scene. Doesn't require that many cells. You leave enough cells behind when you touch something with your finger to give a genetic profile now. They need about 10 cells. And so it's very possible that you touch something and you've left 10 cells behind at a crime scene. So they will find something that someone has potentially touched. They will do a genomic DNA isolation. They will do PCR on the 24 different locations, and then they will start doing statistics. And we're going to learn how to do statistics on Monday, so I'm not going to get into it here. Other than to note, they've now got locations on every single one of the 22 chromosomes to avoid trying to get the maximum flexibility. And how they're able to tell whether it's male or female DNA that's been left behind in the crime scene is from the amylogenin gene. The amylogenin gene on the X chromosome is one particular size, and on the Y chromosome it's a different size. Now for everybody who has an X chromosome, it's all the same size. And for everybody who has a Y chromosome, it's all the same size. But they're not needing to discriminate between what the size is. They're just saying, do we have two different sizes when we PCR up the amylogenin gene? And if we have two different sizes, one comes from the X and one comes from the Y, and it's a male. And if you only have a single size of the amylogenin gene, it's because there are two X chromosomes, and so they're both are going to give the exact same size. So if you only have one band in the amylogenin gene, it's going to be a female because it's XX. There, of course, are XXY cases and weird things like that, but for the most part, we're just going to confine ourselves to XX and XY because that's a whole bunch of different chapters in a book, going beyond that. Okay, so they have weird names to them. They have weird names like TPOX and CSF1PO. So initially, when people were developing all these short tandem repeats, everyone was naming them according to their own little nomenclature. And finally, they got together and said, come on, guys, we need a consistent naming system among all of these. And so that's when they came up with things like D5S, 818 or whatever. So they said D for DNA, 5 is on the 5th chromosome, S, 18, S518 or whatever. That's the particular location on the chromosome where this particular short tandem repeat is found. So now I want to talk about this slide. Okay, so we've talked about PCR. And so let's say someone's looking at chromosome number 5. And they have on chromosome number 5, just outside the potential different number of repeats that everyone has. Because remember, there are tens of thousands of potential places that have repeats. So these were specifically picked for having wide genetic variability in how many people had each of these number of repeats, and for having very consistent locations where the primers can bind. So when you are binding primers outside of these repeats, the repeat region is variable between the samples. So if we're looking at chromosome number 5, you might have 6 repeats, 7 repeats, 8 repeats, 9 repeats, and they're going to be one after the other. But 8 repeats is bigger than 7 repeats. It's going to be a little larger. But the important thing is the place where the primers are binding has to be constant. So for each individual location on the DNA, chromosome number 5, they had to test to make sure that everybody has the exact same sequence on chromosome number 5 so they can make oligonucleotides, which are going to bind to every single person's DNA and amplify in between those primers. Now, you're not amplifying large pieces of the DNA. You're amplifying usually between 300 to 600 bases. So all of these fragments are very small. They're not sequencing the DNA at all. Sequencing is done by companies like 23andMe and Ancestry, where they're looking at single nucleotide polymorphisms in the DNA, but they're not actually, the police department does not sequence the DNA. They're only interested in how long it is. So you might have inherited a 7 from your mother and an 8 from your father, or you might have inherited an 8 from your mother and a 7 from your father. So there's 2 different ways that you can be a 7 and an 8. And that's homozygous, and the 2 different ways is going to play in when we talk about statistics on the Monday after spring break. But basically, you're at each location that they're PCRing, you're either going to be homozygous or heterozygous. If you inherited 2 different numbers of repeats from your parents, they're going to be different sizes, and they can be resolved on a gel because they're going to be slightly different sizes. Something with 8 repeats is going to be a little larger than something with 7 repeats. And if it's homozygous, it meant maybe you got an 8 from your mother and an 8 from your father. So you got the same number of repeats from both parents. So when you are doing this, that's one location on the DNA. But I've said they're doing 24 different PCR reactions all at the same time. So how are they able to do 24 different PCR reactions all in one tube and still be able to separate all of those out? So one thing they do is they change the distance where the primers are binding. You can have the primers binding very close to where your repeats are, and if the primers are binding very close to where your repeats are, you're going to get smaller size ranges of fragments. And if your primers are binding farther away from where your repeats are, you're going to get larger sized fragments. And they can be resolved on a gel because they're going to be different size ranges. So now, I want you to look at this picture that I gave everybody. We're going to talk about that. So you're looking at this picture right here. Okay, so look at panel C that's on the right-hand side. And panel C has a genetic profile for 6 people. So see the numbers 1, 2, 3, 4, 5, and 6. Those are 6 different people that they have done PCR on their DNA. Any lane which has an L has a bunch of size markers. And those size markers are specifically designed to tell them and say, okay, if you have, let's say TPOX, if you have TPOX, if you have 6 repeats, it's going to be this size, the same size as that lowest band in the L on TPOX. For all the L's. All the L's are exactly the same. So the L's to the left, the L's to the right, the L's in the middle, they're exactly the same set of markers. You have markers, 2 people DNA, markers, 2 people DNA, markers, 2 people DNA, so that you always have someone to exactly compare it to. So let's say person number 6 on TPOX, and you wanted to say, okay, how many repeats, if we are amplifying the TPOX location in their genomic DNA, how many repeats does that person have? So what does it line up with? So you can see, 6, 7, 8, 9, 10, 11, 12, 13. So what does person number 6 in TPOX line up with? 8. So they would have, is that homozygous or heterozygous? Homozygous. How many bands do you see? 1. If it's 1, is it homozygous or heterozygous? Homozygous. It's homozygous. So this means they inherited an 8 from their mother and an 8 repeat from their father. Okay? So both parents contributed the exact same number of repeats. And you can, as you look across person number 1 and 2 and 3 and 4 and 5 and 6, you can see 8's pretty common. A lot of people have 8 repeats of TPOX. I have 8 repeats of TPOX. Now let's look at TH01. Let's look at person number 6 in TH01. So same person number 6 on the right-hand side, just down 1 to TH01. How many repeats does person number 6 have at TH01?
6 and 8. 6 and 8.
6 and 8. So does anybody not understand, this is important, Does anybody not understand why it's a 6 and an 8? Don't be afraid to holler. Okay, so that's good. You got to be brave. Okay, so look carefully at person number 6 at THO1, and you can see that size marker. And if you see that size marker, it goes from 5 to 11. And so you have 5, 6, 7, 8, 9, 10, 11. And so you can see person number 6 has little bands next to the 6 and the 8 of the size markers. Do you see that? Okay, so this tells you they're a 6 and an 8. Does that work out for you too? Okay, so then you can say person number 6 is a 6, 8 at this location. And then you can go on and you can do CSF1PO for person number 6, and you can do VWA for person number 6, and you can do, because you are changing the distance that the primers are from the repeating unit of DNA, you're going to get a different size range on the gel. So you can separate out on this gel, you can separate out 4 different size ranges. But wait, she said that it was possible to do 24 different ones all in the same tube and all in the same gel. How is this possible? Well, now look over to the left, to that little black figure. What color are the CSF1PO, the TPOX, the THO1, and the VWA in? Okay, so they're all in red. So that's because they all have a red fluorescent marker on the ends, added to the ends of their oligonucleotide. So you can tell the computer, show me only the red, or show me only the green. So you can be running all of these bands at the exact same time. You can be running 24 different reactions, but if these 24 different reactions have a bunch of different fluorescent markers attached to them, then you can just see a couple at a time. So you can have the red, where you can see the panel on the right. You can have the green, where you can see the center panel. And there's blue, and there's yellow. There's a bunch of different kinds of fluorophores that you can add to your oligonucleotides, and then you can amplify all 24 at the same time and just see a couple of them one at a time to be able to look at them along with the appropriate size markers. Because if you look, the size markers come in red and green also. So the size markers, for VWA will show up if you say, show me red only. And you'll get a completely different type of size marker if you're looking at the greens, which goes for the D6, which is in that exact same size range, but now you're telling the computer, show me only the green. So this idea is called multiplexing, of doing multiple PCR reactions, far more than one PCR reaction at one time. And so when we multiplex, we're actually going to be using CSF1PO, TPOX, and THO1. We're going to be amplifying all 3 at the exact same time on your genomic DNA that you will have isolated from your genetic sample. So I think, yes?
Can you explain how we know if it's homozygous or heterozygous?
So, okay, excellent. So if you look at TPOX and look at person number 6 in TPOX, how many bands do you see for person number 6 in TPOX? 1. So by definition, it's homozygous, because if you have inherited 8 repeats from your mother and 8 repeats from your father, they're going to be exactly the same size. And so we call that homozygous. They're the same size and they cannot be resolved. You don't know. You're just doing the size. You don't know, which came from your mother, which came from your father. It doesn't matter. You're homozygous. But versus THO1, you see 2 different bands there. By definition, you're heterozygous. Okay, I think I will follow all this up on the Monday after spring break, where I will talk about the statistics and tell you things that you want to make sure you talk about in your intro. So you can start working on your intro with some of the things I already talked about, but I'll be making sure that you know to hit the high points. So meanwhile, make sure you're looking in your lab manual, find the oligo stuff, find the isolation of DNA using the DNA IQ system, find native polyacrylamide gel electrophoresis, find agarose gel electrophoresis. There's all these things in there. You're going to be pulling all these protocols together. Okay? Yeah.
How does it know when to add to what?
Because they all have very specific sequences. Because, so remember outside of the number of repeats, you have very specific sequences. So this, all of those are going to bind to this location at
No, it's.
Oh, because it's the
Also,
primer.
yes, she was
the
Has
primer.
the fluorophore.
Yeah, the primer.
Oh, okay.
Yeah,
Oh,
primer.
okay, that's what I, okay, that's how you can.
Right.
Okay, makes sense, okay.
All the,
And then,
all the work
I
that
was wondering, so when they're, extracting DNA, like, for, let's say, like, or how do they do that?
So one of my summertime fun projects was working with Dr. Mahoney in biology. And he had me working on deer scat, which is deer poop. His scientific question was, he wanted to know, so we collected deer poop from different locations. Is this one deer, which is ranging across this huge area, or is each deer staying in a single location? So this poop is different from this poop is different from that poop. So I came up, I helped him with a protocol to be able to isolate poop. So there are actually kits that you can use to get DNA from poop, and you do it very carefully to get the epithelial cells on the outside. So what we did is we dried the deer scat so that it was dry, and then I would put it in a little baggie. I would cut off the end of the baggie, and I would have just, like say, 100 microliters of buffer in the baggie. I'd put a couple of little deer poop balls in there, and I would massage the poop very gently to help get the epithelial cells off the outside of the poop. And of course, you get other stuff besides epithelial cells, sort of, you get brown liquid. But you enhanced it for the epithelial cells. And then you're putting it through columns, kind of like the DNA IQ kit, but a little bit different. So you're doing a DNA cleanup kit so that you can then do PCR on it and try to identify the deer using STRs, deer lines, or for deer rather than for humans. And you can do this for dogs. So when people's dogs leave dog poop at a particular site, and there are buildings, if you want to move in with your dog, you have to submit a genetic profile. You have to submit your dog poop, and they will, and with a $600 fee or something, they will get a genetic profile for your dog. So if poop is left behind on the sidewalk or something, they will collect it. They will send it off to the company to say whose poop is this, and when they find out it's your dog, they will charge you $1,000 for leaving poop on the sidewalk.
Yeah, because I took forensic anthropology, and then like we went over this. So this why when I saw this slide, I was chuckling because I'm like, oh, I remember that.
Because I only gave a lecture.
No, it was
Was it Erin Mays?
yeah.
Yeah, she always puts.
Dr. Mays, yeah.
Yeah, so she puts my lecture up. So.
Oh, so that was your lecture. That's why I was like, oh, that looks so familiar.
Yeah.
And the guy with the poop in the living room.
Yeah. So yeah, that's the lecture I give her. Although in like so this semester, for example, I couldn't make it because I had this class.
Oh.
And it was